All right, class. Thanks for tuning in. So the game plan today is I'm going to finish Chapter 4, Part 1, and we were looking at the structure of the nucleus. So I'm just going to back up a couple slides here. And then we're going to transition into the endomembrane system. And then we're going to see how far we can get. So I'm just going to set my watch for about 35 minutes. I'm just going to lecture for 35 minutes, and then we'll finish it Wednesday. And we're looking at a quiz on Thursday. On Google Classroom, I might say a quiz on Wednesday, but no, just keep that in mind that it will be on a Thursday. So the nucleus, we discussed, has a nuclear envelope on the outer, outer layer, if you will. It's made up of two phospholipid bilayers, and it is continuous with the endoplasmic reticulum, which is kind of cool. The nuclear envelope also has small little openings called nuclear pores that allow things to move freely between the cytoplasm and the nucleoplasm, which is basically inside the nucleus. There are basically passage is restricted to two things: proteins moving into the nucleus to be incorporated into some nuclear activities, and then RNA or RNA protein complexes being exported to the cytoplasm. And then we have, the inner surface. If we look at it, we can see that there's a very vast network, fiber network, I should say, going on here. I showed you that trippy video of how the nuclear pore complex can bring things into the nucleus using these tentacles, and it kind of dunks it through the basket, if you will. So there's that trippy video. We're not going to watch it again. Chromatin is also found inside of the nucleus, and it has to do with how DNA is packaged. So chromatin is DNA, and I like this diagram here because if you just look at the double helix of DNA, this is what it looks like. But then if you were to supercoil it, twist it again, repackage it, and basically wrap it around some proteins, now you got some chromatin. And then if you were to coil and twist it again, now you'll get some packed nucleosomes. And you keep coiling and twisting. Eventually, you're going to make a chromosome. Okay. DNA contains the hereditary information specifying cell structure as well as function. And so remember in prokaryotes that they don't have a nucleus, but they do have a kind of condensed genetic area. Another thing about prokaryotes is that their chromosome is singular. Okay, so it's like continuous. It's like a ring. It's circular. It's one ring. Eukaryotes, we have multiple linear chromosomes, like humans have 23, and carp have 104 chromosomes. So they look like X's. We call that linear. But in prokaryotes, it's circular. Okay. Chromosomes are organized with proteins, and so we call that chromatin part. Proteins are called histones. And so when DNA is wrapped around histones, a nucleosome is formed. So to recap a lot of vocab here, chromatin is DNA plus proteins. Okay, and those proteins are called histones. When you have DNA wrapped around histones, you get the nucleosome. So hopefully you got that. DNA also has the ability to loosen or extend, so that regulatory proteins can attach to specific sequences and to compact into highly condensed form during cell division. And so there are times when parts of the chromosome will kind of uncoil a little bit and allow proteins to bind. And so this happens when genes get turned on. But then during cell division, your chromosomes condense because they don't want to lose any bits of themselves during the process of moving and splitting in two. And so they get highly compacted. And that's why you can see chromosomes during cell division, but not, during the process of interphase. Okay. So, with life science, I always tell the kids if you see inside the nucleus, like if you're looking at a picture and you see it kind of look like spaghetti, well, you're looking at chromatin. All right, continuing on with the nucleolus here. The nucleolus is responsible for ribosomal subunit manufacturing. So this is where basically parts of ribosomes are made. So ribosomes make proteins, and a cell needs a lot of them. There are ribosomes everywhere inside the cell. So there are hundreds of copies of genes that code for ribosomal RNA, or rRNA. You may see that term. And so they get clustered on the chromosome. Another part inside the nucleolus is the nuclei. ribosomal assembly areas within the nucleus, a very dark, stained area, very easily seen with a microscope. Basically, it's just saying this is where a lot of ribosomes are getting put into place or are being made. So remember, our ribosomes are the cell's protein synthesis machinery. So it's composed of two subunits of our RNA, or ribosomal RNA. And after you make ribosomal RNA, it requires two other forms of RNA to make proteins. Your mRNA, which carries the message from DNA to the ribosome, saying these are the amino acid sequences that we have to put in order. And then what carries those amino acids to the ribosome is transfer RNA, or tRNA. I just love how they're so original with naming it, which is nice. Transfer RNA, tRNA, because they carry amino acids to the ribosome. Okay, ribosomes, they can be found free in the cytoplasm, or they can be bound to, like the ER. So free ribosomes are usually found in cytoplasm, nuclear proteins, or even mitochondrial proteins. Okay, and then also ribosomals are universal machines. They're found in all three domains. They just have a slightly difference in size between eukaryotic organisms and prokaryotic organisms, as well as archaea. Okay, so that was Chapter 4, Part 1. All we did was discuss why, cell theory, 3 statements, scrolling down why cells are small, what's the problem with magnification and resolution, difference between gram-positive and gram-negative. Bacterial walls have peptoglycan. Flagella is used for movement, and then we spent this last part talking about the structure of the nucleus. Now, part 2, we are going to look at the endomembrane system. So, section 4.4 and 4.5. So, the endomembrane system is basically anything that, has a membrane. And so we're going to take a look at the eukaryotic cell here, because eukaryotic cells have a lot of membranes or organelles that carry out important functions. So divides cells into compartments, channels molecules through, may provide surface for the synthesis of lipids and proteins. So it's just jam-packed with different kinds of membranes. So this is a major difference between eukaryotics and prokaryotes. So the largest internal membrane found in a eukaryotic is the endoplasmic reticulum, or the ER, which stands for within the cytoplasm, or a little net. And the ER is also made of a phospholipid bilayer embedded with proteins, and those proteins are ribosomes. And we'll talk about the differences between smooth ER and rough ER. So a little preview of the endomembrane system. So here I have my nucleus, okay, and this is my ER, okay, and it says nuclear envelope is connected to the rough ER, which is also continuous with the smooth ER. And so the rough ER is ER that has ribosomes embedded into it, and the smooth ER has no ribosomes. And the difference is rough ER will make proteins, and the smooth ER will make lipids. Now, step 2 says membranes and proteins produced by the ER flow in the form of transport vesicles to the Golgi apparatus. And so proteins get made, they get packaged, they get packaged into these little vesicles, and these vesicles float around in the cell. Okay, I shouldn't say they float. They get tagged for a destination. So in this case, they will go to the Golgi apparatus. So they enter the Golgi apparatus, and they fuse through the process of endocytosis. Now these proteins get modified and repackaged until it reaches the end of the Golgi apparatus, where different fates, can arise. So here it says Golgi pinches off transport vesicles and other vesicles that give rise to lysosomes and vacuoles. So it can help, maybe these proteins are destined to become enzymes that break things down. So they'll become the garbage bin of the cell, or the storage people. But lysosomes break things down, vacuoles store things. Lysosome available for fusion with other vesicle for digestion. So then when it's active, it will fuse with maybe a protein that needs to be degraded and basically eat it, not eat it, but recycle the parts. Transport vesicle carries proteins to plasma membrane for secretion. So these things leave the cell. And, communicate or send a message to different cells. And then final thing here, plasma membrane expands by fusion of vesicles, and then proteins are secreted from the cell. So here's the process of exocytosis, where it fuses with the plasma membrane, releasing the cargo. Now the ER weaves and creates channels and folds, and so basically they're like compartments, but we call these compartments cisternal space or the lumen, and that is labeled here. So this orange area is considered the cisternal space. Remember the cytosol? We talked a little bit about the cytoplasm, how it contains dissolved organic molecules, proteins, as well as ions. Well, the cytosol is the region exterior to it. And it is not listed, but it would just be just out here. Okay, so the ER, there's two types of ER, rough ER and smooth ER. So the rough ER is a site of protein synthesis, and it's rough because of those ribosomes. Proteins get made, and they are exported from the cell, either or sent to lysosomes, or sent to a vacuole, or they get embedded into the plasma membrane. Proteins can be modified by adding carbs, and then as a result, instead of just having a protein, you get a glycoprotein. And a lot of times, glycoproteins are destined for secretion. So this part right here says these proteins enter cisternal space of ER first. Well, remember, it all starts in the nucleus. Okay, mRNA travels to a ribosome. Maybe it's the ribosome found on a rough ER. The protein gets made, gets packaged into a vesicle. And it's just saying that it's entering the cisternal space of the ER first. Well, this would be the cisternal space. Enters, packages through, leaves. So, yeah. Proteins are packaged into vesicles that involves the Golgi apparatus, and then the ER also makes membrane proteins that are embedded into its own membrane. So those are the responsibilities of the rough endoplasmic reticulum. Now the smooth ER also has several roles. Doesn't really have a lot of ribosomes. That's why we call it smooth. And so instead of having these nice little folds, we get a network of tubules, basically. May contain some embedded enzyme that catalyze the start of carbs and lipids. And this is also where calcium is stored in cells. And calcium, when we talk about neuron signaling later on this year, acts as a signaling molecule. So it can communicate with other cells. We use this for muscle cells during contraction, and then a signaling pathway. Ratio of smooth ER to rough ER does depend on the cell. So cells that are found in the testes, intestines, and brain usually produce more lipids. And then cells where proteins are secreted, like antibodies, immune system, they'll have more rough ER than smooth. Another role for the smooth ER is modification of foreign substances to make them less toxic, or detoxification. So vesicles might fuse with the smooth ER and get modified so it's not as toxic. So it's a less harmful form of what it originally was. So it looks like I have a video here, and I need to approve it. Give it access, yes. I hope it works. Well, this is not good. Oh no. I have the spinning wheel of death. Awesome. Awesome. I can't even get out of PowerPoint. Well, class. I don't even know what to say right now. Do I want to wait or quit? Let's just quit. Let's just quit while we're ahead, right? Okay. Okay, PowerPoint, I'm going to give you a second chance here. Actually, no, I'm just going to force quit. Just quit. Just give up. Told you to give up. I guess I can clean this stuff up, huh? All right. I'm so sorry, guys. What do I want to do? I want to open recent. So it looks like the video is the thing that's tripping it up. So I think I'm just going to move on. And then if someone reminds me, I'll be happy to play it. Okay, so the Golgi apparatus, its main purpose is to sort and package proteins. So it gets these vesicles usually from the ER and will repackage and redistribute to its final destination. Golgi apparatus does have a front and a back, and so the front is the receiving end. We call this the cis face. It's located near the ER. That's the front end. So materials move to the cis face, and these vesicles, they bud off from the ER. They fuse with the cis face, move to the interior. Remember, we call the interior the lumen, and they continue to pass through until it reaches the very end of the ER, and we call this the trans face. And then at the very end, it's discharged as a secretory vesicle with a final destination. Is it going to become a lysosome? Is it going to become a vacuole? Will it be secreted from the cell? Will it leave the cell and go elsewhere? I lost my mouse. You guys see it? It just froze again. Shoot me. Just kidding. Just kidding. Well, you guys want the good news? We are at 17 minutes and 40 seconds from when I started my watch. Oh, come on. It's just one more reason. One more reason. Herman Norcross, can you hear me? I think I would take an ITV problem right now versus this. Come on, Microsoft Windows. Pop up another window. Ah! Oh my gosh, we're back. Okay, you know what? I'm just not going to go into presentation mode. Okay. So here's the Golgi apparatus. This is showing a kind of a 3D model of what it looks like. The cis face, which is facing the ER, and then the trans face, usually facing the plasma membrane. So they're outgoing transport vesicles. Here is the kind of like a 2D model of its cartoon diagram showing that vesicles come from the endoplasmic reticulum, fuse to the Golgi apparatus, and it will either become a secretory vesicle, it will secrete outside the cell, might become a part of the plasma membrane, might become a lysosome. Inside each part, if you will, lumen, there are different enzymes that work on it. So remember, these proteins come in, they kind of get repackaged, modified a little bit, and then targeted for their end destination. So proteins and lipids made on the rough ER and smooth ER are transported to the Golgi and then modified as they pass through it by all these different enzymes that either, remove manganese, I think that's what it is, I'm not sure. Addition of glycoproteins. So addition or modification of short sugar chains, glycoproteins, glycolipids. As a result, newly formed altered glycoproteins lipids collect at the ends of the Golgi bodies in the flattened stacked folds called cisternae, and then the cisternae will push together and pinch off with the glycoproteins or lipids, and these vesicles fuse to their locations or final destinations. So the Golgi can also make cell wall components as well. All right, switching gears. So one destination that a vesicle can become after it leaves the Golgi apparatus is a lysosome. And I always like to call the lysosomes the garbage men of the cell. They contain digestive enzymes. And inside of the lysosome, we have some very dangerous degrading acidic enzymes that can break down proteins, nucleic acids, lipids, and carbs. So lysosomes go around and they break down old, damaged organelles and recycle all the molecules and bits. But they can also eliminate other cells that the cell has engulfed by phagocytosis. So here we have a lysosome, and it will fuse to like a food particle and break it down. Digestion organelles, this mitochondria, has gone past its life expectancy, so the enzymes fuse, break it down, repackage, recycle all those parts. Okay. One thing I like about this diagram is it zooms in on the plasma membrane of the lysosome, and you can see that it is a lipid bilayer. So think for a couple of seconds here. Why do you think it's a lipid bilayer instead of just one layer of lipids? All right, here's the answer. This stuff inside the lysosome is potent. Okay? If it leaks, becomes damaged and releases this hydrolytic enzyme mixture inside the cell will die. So the cell wants to protect itself from it by using a lipid bilayer. So double the protection. Okay. I want to talk a little bit about microbodies. Okay, these are something that have kind of come into play just recently. Very diverse. These guys carry enzymes. Okay, they are enclosed in a vesicle. You find them in plants, animals, fungi, as well as protists. It's not a lysosome. Okay, these guys bud in the endomembrane system. That's what lysosomes. That's where they come from. But microbodies grow by incorporating lipids and proteins and then dividing. So they have different origins of how they are made, but they still carry some pretty potent enzymes, if you will. So one example of a microbody, peroxisomes. Spherical and oval shape, contain the enzyme catalase. Ding, 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 ding. That should be ringing some bells. Catalase protects the cell from toxic effects. And so we looked at catalase in liver. Okay, so that was an example of a microbody, peroxisomes. Lysosomes, on the other hand, we categorize them as hydrolases. Some other examples of microbodies, or I should say lysosomes. Yeah, hold on, I'm just going to go back here. 1, 2, skip a few, 3, 4. Okay. Glyoxysome. We find these guys in plant cells, and they contain enzymes that convert fats into carbs, which is pretty important. And then peroxisomes contain enzymes that catalase the removal of an electron and a proton. So hydrogen peroxide is produced by the byproducts of some type of activation. And so it contains catalase and breaking down H2O2 into water and oxygen, something that is less harmful. All right, some other organelles you need to know. Vacuoles, and they store storage and water balance. So the shape of the central vacuole will kind of determine whether or not the plant is wilted or if it's turgid. So the membrane surrounding vesicle, we usually call it tonoplast. You can see it labeled here. And it expands or shrinks dependent on osmotic balance and conditions. In plants, we do have a central vacuole, but there are different vacuoles found in different cells. So keep that in mind. Animal cells do have vacuoles, just not a central vacuole. Okay, continuing on with the endomembrane system, we're going to switch gears and talk a little bit about energy and how cells get energy from within. And so they use these two organelles, dependent on the species, the mitochondria and the chloroplast. Now, mitochondria is found in all cells, animal cells and plant cells. Common misconception, people just think plants have chloroplast, but that is not true. So plants undergo photosynthesis as well as cellular respiration. Okay, plants have chloroplast and mitochondria. Animal cells do not have chloroplast. They have a lot of similarities. They both have a double membrane layer, as you can see here, double membrane, and they each have their own DNA and protein machinery, which is kind of odd. So we're going to talk a little bit about that here. Function involved in energy metabolism. So here we're going to take in glucose and oxygen and release some carbon dioxide gas and water, as well as some ATP. And here we're going to take in sunlight, water, and carbon dioxide, and we're going to make a carb, glucose, as well as 6 molecules of oxygen. Okay, we haven't got to this part yet, but we will. This slide is showing kind of how energy is used in mitochondria versus like the chloroplast here. So we haven't talked about this, but in cellular respiration, how it makes ATP is through this protein called ATP synthase. And ATP synthase, what it does is it takes ADP, which is adenosine diphosphate, and combines it with another phosphate. So then you get ATP, which is adenosine triphosphate. And so this ATP synthase is basically like a water turbine, okay? So think about a turbine for a second, okay? Water flows through it, and then these things, the paddles, if you will, spin, and you can generate electricity. Very similar. But instead of water or even wind, we have hydrogen ions that flow through, causing the paddles to spin, and then combine an ADP and a phosphate to form ATP. And we can make a lot of ATP this way. So then how it works is you have to build up a concentration gradient of hydrogen ions outside, if you will, so they can flow in and spin the paddles to physically combine ADP and P. And so we use an electron transport chain to drive hydrogen ions outside, and then use that as an electrochemical potential gradient to drive ATP synthesis. Now, chloroplast, you see something very similar. Okay, here their ATP synthase is not inside, it's outside. And so light and a few other enzymes that we'll talk about later on, their main purpose is to get hydrogen ions inside to store it as an electrochemical potential, and then make it go through ATP synthase as it leaves. So same kind of machinery, if you will, just a little bit, different. So let's focus on the mitochondria here. Mitochondria, what it does is it metabolizes sugar to generate ATP. We usually say it's a kidney bean shape, tubular, or maybe sausage shape, about the size of a bacteria. And I italicized that because that's pretty important later on here. It's found in all types of eukaryotic cells, and it has 2 membranes, a smooth outer layer, denoted by this darker brown color, and then inner folds with many contiguous layers called the cristae. Okay, so there's the cristae, light tan kind of color. Cristae divides mitochondria into 2 compartments, the matrix and the inner membrane space. So the matrix looks like the water, okay? And then the inner membrane space would be outside of that, so like the gap here, and the gap up there. They have Proteins on and embedded that carry out oxidative metabolism, and they also have their own DNA with genes that code for their own types of proteins. So it's like a cell that acts within, another cell. It's not fully autonomous, though. It is dependent on the cell that it lives in. If that's kind of odd. Eukaryotic cell does produce new mitochondria when it divides, and so the mitochondria actually divide themselves into two. So it will grow and elongate, and then when it gets big enough, it will pinch off into two new mitochondria, one for each new daughter cell. Now, chloroplasts, they use light to make ATP and sugars, so they make their own food. This is a pretty good advantage for those that are photosynthetic, with the use of the pigment chlorophyll. It also has two membranes, a larger and more, but it's a little bit more larger and complex than the mitochondria. So here we have the outer membrane, the inner membrane, and then we even have some more folds. So we'll talk a little bit about the thylakoids. When you get a bunch of thylakoids together, you get the grana. So the grana, closed compartments stacked, each contain a few or several disc-shaped structures called the thylakoids. On the surface of the thylakoids is where you will find photosynthetic pigments. The stroma is the fluid matrix that surrounds the thylakoids. If I had to say, it would be this kind of light green kind of color. Chloroplast also contains its own DNA. If there's another type of plast out there called the leucoplast, which lacks pigment, maybe it's stored in starch, so we call those amyloplast. Chloroplast, leucoplast, and amyloplast, we call them plastids, and it's just something that your book just mentions. I'm not going to highlight too much on it on the exam. But the fate of what does it become? So kind of like with the endomembrane system, the fate of the vesicles. Well, same here. Here we have a proplastid, which is kind of like the stem cell of a plastid of some sort, where it can become a chloroplast, a leucoplast, or eventually an amyloplast. So an amyloplast is actually part of a leucoplast, just stores starch. So here's a nice little flow chart, and I'm sure that each one has its own little chemical signal pathway to determine what it will become. Okay. This diagram up here is going to talk about how plastids evolved. So they're saying that there was a cyanobacterium, okay, and it had a chloroplast, and the chloroplast underwent some changes. Maybe it specialized in different colors, or maybe it specialized in just storing starch, or light-dependent development, or even just development in itself. And so possible ways that's how plastids arose. But going back to mitochondria and chloroplast and their similarities. So these organelles both have their own DNA, both kind of replicate on their own, okay, both have a function dealing with energy, okay. So I'm going to talk about a theory called endosymbiosis. So symbiosis means there's a close relationship between organisms of different species that live together. And so when you have endosymbiosis, basically we're saying that they kind of live inside. So today's eukaryotic organelles evolved by symbiosis arising between two cells that were each free living. So here's this early cell. Maybe this is what it looked like. I don't know. And if it had DNA, with a nucleus, it became eukarya. So this is what is shown as the nucleus, how the nucleus formed. But then we also have bacteria and archaea, who, some picked up the ability to capture light and make its own food. Huge advantage. Some were not photosynthetic, but they had, they were basically like mitochondria-like, and could metabolize things around them to create ATP. And so what they're saying is that this bacteria or this archaea was swallowed by an early cell that had a nucleus, and they lived in harmony within each other, okay? Or I should say the mitochondria lived in harmony within the cell that swallowed it. And they, each gave each other something and benefited from each other, and now they are just together. So that's the theory of endosymbiosis. So one cell, a prokaryote, engulfed by and became part of another cell. And then the engulfed prokaryotes provided host with special metabolic abilities. And then two key eukaryotic organelles that are descendants of endosymbiotic prokaryotes are the mitochondria and the chloroplast. Why this hypothesis is supported, like what's the evidence behind it? Both mitochondria and chloroplast are surrounded by two membranes because when it got engulfed, it got the inner membrane from the plasma membrane and then the outer from the plasma membrane or ER of the host cell. So that's why these guys have two membranes. Mitochondria are about the same size of a prokaryote. The cristae resemble these folds that we're going to talk about with bacteria. How do bacteria get away with carrying out photosynthesis yet they don't have organelles? Well, they use their plasma membrane, and their plasma membrane folds and captures pigments, holds pigments so it can capture light energy. So that's how they do that. Mitochondrial ribosomes are similar to prokaryotic ribosomes in size and structure. Mitochondria and chloroplast have circular molecules of DNA, just like prokaryotes. So they're not linear like eukaryotes, they are circular. The genome of mitochondria and chloroplast are similar to proteobacteria as well as cyanobacteria. And then also mitochondria divide by simple fusion, where they grow, elongate, and just split in two. Okay, I have two more slides here, and then I'm done lecturing. So sorry about wasted time with the technical difficulties. So this diagram is just showing mitochondrial DNA versus chloroplast DNA. Cartoon diagram with the transmission electron microscope. So the electrons are traveling through the specimen, giving you a 2D image. And you can see that there are lots of inner folds in here. Here's the stacks of thylakoids forming your guarana. So there is a video here by Mr. Anderson, pretty good guy on YouTube. You can look it up. He talks about the endosymbiotic theory, hypothesis, if you will. And I'm just not even going to bother because it's probably not going to play. But I will wrap up Chapter 4 tomorrow. Continue to work on your guided notes. Quiz on Thursday with organelles. There's a table in your book. Please, 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 please look at it. All right, see you tomorrow.